request forms, handbook, clinician's handbook, as well as POCT. And also will handle requisition of blood and blood components. We will also handle laboratory errors. So the panelists include Dr. Francis Ndiangui, the HOD, Department of Laboratory Medicine. We have Peter Kiptim, in charge clinical chemistry laboratory. Dr. Makori, who will be handling requisition of blood and blood components, is a medical specialist, hematology. Then we have Dr. Kasim, who will handle POCT. Since we are constrained with time, we'll call upon Dr. Francis Ndiangui to give opening remarks as we proceed. Dr. Francis Ndiangui.
Thank you, Mr. Mukaya, and good evening to all the attendees of this evening's webinar. I'm Dr. Francis Maina Ndiangui. I've introduced. I'm the head of department, laboratory medicine, Kenyatta National Hospital. Mine is to say that we are glad and privileged to present this sensitization to our clients and also clinicians as well as staff in the hospital. And basically is to allow us to interact with our customers, who are the clinicians, the patients, our staff. And this webinar is just basically for that interaction between the department and the clinicians. As part of the standard requirement, ISO standard 15199 laboratory accreditation, we are supposed to have at least interaction with our clinicians in terms of to advertise on what we do and advise on what laboratory is all about on all aspects, including patient laboratory, how to fill the request form, how to take the samples, what is required in the acquisition of samples, our laboratory, various laboratories, where they are and what they do, and also even what are the charges to our clients in terms of the services that we offer. So basically that's the interaction today. And as he has outlined the presenters, that will come up will be covered in the 4 sessions that we have. Back to you, Mr. Mukaya. I don't think Mr. Mukaya is still in the call. I think in the program, I'm the first to present. Maybe you would allow me to share my first presentation. I hope my presentation is visible and the slides moving.
Yes, they are.
Yes, so the first presentation is the laboratory request form. And I'll just start by noting that what we say, there's the notion of brain-to-brain loop for laboratory diagnostics, meaning what the clinicians first thinks of the request to make in the brain, then communicates that to the laboratory, then back the results are brought and back to the clinicians and makes a decision for the management of the patient. So the first step in this loop involves the selection of the appropriate laboratory tests in the brain. Then there is the communication through the laboratory request form, as I has highlighted. I think this is very important step in the process, and that's what we dwell on in the presentation. Then there's intermediary steps, such as identification of the patient, specimen collection, and specimen handling, as well as specimen analysis in the laboratory. Then there is the release of the results for the patient, for the physicians to review, and a reaction to the same laboratory information or laboratory report, interpret the results, then implementation of the clinical decisions once it goes to the clinician. So today we take the most, just highlight one aspect of it, the laboratory request form. What is the importance of this laboratory request form? So request form allows for the communication between clinician and the laboratory. And this usually acceptance of the request form is considered an agreement with the patient or the client. Once you bring the specimen and the request form is filled, once we accept, then we form an agreement that we are going to process the sample and you are going to give a report to that whatever has been requested. So this request form, it is mandatory to the minimum that it should be unequivocal, have unequivocal traceability of to the patient and to the sample itself. Each can stand on its own, that you have a request form and you are able to trace back to the patient and to the sample itself, so that there's proper identification to avoid errors or mistakes being occurring in the whole process. At least it should have identity and contact information of the requester, and also identification of the examinations that have been requested by the clinician. Those are basic minimum that should be contained in that request form. So we say that the request form should be formatted in a way that it persuades clinicians to provide all information that could be relevant for correct laboratory testing and interpretation. I use the words persuades in quotes because I think for any information that you want from a person, you need to make it easy to be filled and to achieve the purpose. So that would be the way that you would normally format a laboratory request form. And it should include at least the following. So we have the patient's mainly demographics. So that is the details, the name, address, telephone number, birth date, gender, etc. So these should be all included because they all determine or they're important in the interpretation of the laboratory report. Then requested details. After we analyze, we may want to contact where the result will be sent, and also we may want to request the person or have a query to the person who requested. So the primary clinician who requested at least the details. Then type of primary sample, what sample has been collected, or what sample do you want us to collect for at the lab when the patient comes to the lab, if it's being collected at the lab. Then examinations requested. It has to be clear what are the examinations or what are the tests that want to be done on that specimen. Then clinical information to the laboratory. The relevant information that is required for interpretation. Then date and time and place of sample collection. Then you have the date and time of receipt of the sample at the laboratory. Although this may be substituted by having just a stamp in the laboratory to indicate when the sample was received in the lab. And also it should include any other relevant information. So in the aim of making the laboratory request form easy, friendly to fill and serve its purpose, sometimes you find in an institution, you may have the necessity to create several request forms, because several areas or several laboratories, depending on the test, may require different, may have a different format of reporting, or you may require different clinical details for that purpose. So you may find, like in Kenyatta National Hospital, you may find you have a general request form. We have one that is used in the surgical pathology. You have one in blood transfusion. So all those are different, but basically is to allow us, as I said, to make the form friendly, to persuade the clinician is able to fill that and to serve that purpose of, for that particular test that is being done. Just an example for this is a surgical pathology request form that used in Kenyatta National Hospital. And what you find, you have at least the patient's demographics, you have name, age, hospital number, the ward where the patient is, and all these details are included in the request form. So the clinician is supposed to fill also where the specimen, who took the specimen site, any clinical or relevant clinical history to be included. I'll just take for a scenario where in pathology you require more, that we may want to know how many was the patient had previous biopsies, because it's very important as we report, we may want to review the previous biopsy, because it makes it easy to correlate and also to fasten the process of the diagnosis. Remember, a patient may have several biopsies taken at different times. Reporting one without knowing whether how the other one was reported, we may delay the report or have two reports which are not tied up. So this is the one for surgical pathology. You find now the general one for the other labs, that's the chemistry, immunology, hematology. It's a bit different, because this one is more for able to tick what you want to, the request you want to, or the test you want. And they are arranged more into each section of the laboratory department. So those are two different ones. There's one for transfusion, I think will be referred one by one in this presentation. So what are the challenges or what is the take-home message for this presentation from our standpoint as a department? Is that we have found a lot of issues with the filling of the request form. It is not properly filled, which one it is an issue to us and to the patient, and sometimes it does not, we are unable to complete the report. So most of the challenges that we find is that you have an incomplete patient information. Either you have you don't have where the patient, sometimes age, sometimes where the patient is in terms of the ward. You may have unclear test selection, like the one for general or request form. Then you find a clinician's it allows for tick. You can tick, you can circle, but sometimes you find the tick process may be two tests, but you can see clearly probably that's not what was the intention. So we urge clinicians, when you are trying to request for the test, be clear as possible so that you avoid maybe overcharging the patient if you are requesting a test which is not indicated, because we are not able to clearly know which test is requested. Then incorrect specimen collection details. We don't know when the specimen was collected, maybe even the site. Maybe you have a fluid, it's not indicated which site it was collected. Then missing clinical details, missing clinical details, clinical information which the laboratory person may require is not available. An example is where you have a patient, especially in histology, you may have a patient who has been done, maybe is on orthopedic, has a bone lesion, and you don't have radiology findings. and you find for you to report a bone lesion in a surgical pathology, the radiological findings are indispensable. It's not possible to report without the radiology findings. So if that is missing in the clinical history, it may delay the clinical report because you will have to wait to call and look for the file of the patient for you to get that information, which can be provided earlier if the request form is well filled. Yeah, then we have illegible handwriting. We all know the joke behind the doctor's handwriting. It is not a joke. Usually, it's really in the laboratory. Sometimes the handwriting is illegible, and you are trying to figure out what was this requested or what was this clinical information. So try to be legible as much as possible in terms of when you are filling the request form. Then that if there is lack of awareness of the available tests. So sometimes the clinicians may not know which are the available tests, so you may not request. So that's why in our later presentation, we'll talk about the clinician handbook. So what we say, if you have this request form which is not well filled, then there is a danger that it may cause delay in the testing of the report, in the testing of the specimen, or reporting of the procedure. Then there is an accurate results. It may result to an accurate result because interpretation may be difficult, and a potential harm to the patient. So it is part of what may cause a clinical, it may cause an error in the laboratory report. Then sometimes you may need to reject the specimen if it's not well filled. As we said in the beginning, the request form, the specimen, and they should speak to each other. You should be able to each to stand on its own and to be objectively identified. And if it doesn't occur, then you may sometimes reject the specimen, and that takes more time for another specimen to be collected. So basically, this is this presentation, and I come to the end, and I hope I have highlighted on the importance of having the request form to be well filled for us in the laboratory. As I alluded earlier in my remarks, as part of the ISO certification, or ISO 15189-2022, the laboratory standard, part of what is audited is how well are the clinicians filling the request form. And of course, sometimes a request will be picked and you find it is not well filled. So our request, so our just to urge our clients, our clinicians, is to always be filling the request form fully as it is. We try to simplify it so that we only have the relevant details important for the investigation and for correct interpretation of the report. Thank you.
Muted.
Okay.
Not handle it from my end. The next presenter is Peter Kiptim on laboratory errors. As I had said earlier, we'll handle the issue of concerns and clarifications at the point of plenary. So we move to the next presenter, Peter Kiptim.
Thank you so much, Mr. Mokaya, for this. As already introduced, my name is Peter Kibet Kiptim. I'm the officer in charge, Clinical Chemistry Laboratory, Kenyatta National Hospital. So just a few moments while I share my slides.
As you come in, Peter Kiptim, be conscious about time so that we have some minutes at the end for for the participants to share their concerns and seek clarifications. Thank you.
Thank you. So I hope you can confirm that I'm on slide share and my slides are moving.
Yes.
Thank you so much. Thank you so much, Mr. Mukaya, for the kind introduction. So my name is Kiptim. I'm going to take you through this presentation this afternoon. And the title of my presentation is Understanding Laboratory Errors, the Identification and the Possible Corrective Actions. So this afternoon, this will be the outline of my presentation. I'll just go through the background of diagnostic or laboratory errors. Then I'll briefly go through the laboratory testing cycle. Then after that, we'll just go through the 3 phases and identify and possible corrective action for the errors that occur in the 3 phases. And then I will summarize by just depicting what are the error rates in the laboratory testing cycle. So there are 4 major legitimate reasons for ordering laboratory tests, and there could be more, but it's just a subdivision of the 4. So we order tests for the purpose of screening, the purpose of diagnosing, the purpose of monitoring, and also for the purpose of research. And to achieve this, the laboratory personnel usually apply rapid kits to highly sophisticated laboratory equipments or analyzers. Testing can be done in a conventional laboratory, as we all know, and more recently, there's been a very high towards the point of care, which also laboratory testing can take place. To achieve this, we have several subspecialties. I've just mentioned the core ones. That is the clinical chemistry, hematology, transfusion science, microbiology, parasitology, histopathology, immunology, and molecular studies. The importance why we need to understand this subdivision of laboratory is that because by nature of what these subspecialties do, their procedures are quite different. And if they are quite different, then the procedures and the cautionary methods and where errors could possibly occur would be different. And that's why we specifically need to know which lab, which processes. But in my presentation today, I'm just going to look at the general aspects of laboratory testing. So in an overview of the laboratory errors, in 2015, there was a report that was produced by National Academies of Science and Engineering in the US. And I quote, they say, it is likely that most of us will experience at least one diagnostic error in our lifetimes. And sometimes this would have a devastating consequence. Diagnostic errors cause more serious harms to patients than any other type of medical error. That was according to that report. And in summary, they said, or they concluded that diagnostic errors are very common, they are costly, and they are harmful. So looking at the laboratory scenario, we all appreciate that there's been a huge revolution in the past decades in the laboratory. First of all, we moved from the traditional clinical science to now an scientific enterprise where we have things like biomarker discovery and development and introduction of innovative analytical technologies into practice. The other change that took place majorly is the organization of clinical laboratories. Now we are accommodating 2 apparent opposite trends. So we have the consistency of central testing in the conventional laboratory, and now there is a decentralized point of care options available. These new challenges have raised and have raised the number of errors that we experience in the laboratory. So by definition, the first definition that we got for laboratory errors is diagnosis. This is according to Boninet in 2002, is that a laboratory error is a diagnosis that is missed, wrong, or delayed, and as detected by some subsequent definitive test or finding. And later this was adopted by ISO Technical Report 22367, that a defect occurring at any part of the laboratory cycle from ordering tests to reporting, interpreting, and reacting to results. So basically, what is this laboratory testing cycle? So the laboratory testing cycle usually is a 9-step procedure, and the first speaker, Dr. Diangui, has really handled the first one. So there's always the decision to perform the test, then an order is placed, then the order is transferred to the lab, then identifying the information entered, specimen is obtained. We analyze the specimen, we generate a report, then the report is conveyed by the clinician, data is interpreted, and the clinical response to the results is done. So in this, we divide it into 3 phases, which is the pre-analytic phase, the analytic phase, and the post-analytic phase. So it is important to note that errors in the laboratory are very heterogeneous in nature, as it involves various complex procedures and variety of persons performing all the processes, starting from the ordering of the test to the reporting of the result. And this will ultimately influence the patient care. So as you can see, the 9 steps will be so many people will be involved. So that's the reason why it's a very heterogeneous in nature. So if we look at the pre-analytical phase, this can either occur within the laboratory or outside the laboratory, but majority are usually outside the laboratory. And they are classified into 3 groups. One of them is a physiological factor. Physiological factors here look at the physiology of the patient. If you have to prepare a patient, for example, whether is in a fasting state or the test that you're going to do is cyclic dependent, for example, fertility hormones, or they have a diannual variations, for example, cortisol. It is important to note those factors so that when you're preparing the patient and you're taking the samples at that point, you get to know that these can cause laboratory errors. Then there are errors that are associated with specimen collection, either difficult phlebotomy or too much application of a tourniquet for a prolonged period, a patient being collected sample, whether is lying on the bed or sitting. Those are the issues that might take place in the specimen collection. Then we have interference factors, and these can either be drugs or any other thing that the patient is taking at that time. Then other factors to consider are the test requisition that has already been covered. Then there is the positive patient identification, so that the sample that you're going to collect is positively attached to the patient that you're requesting the test. And then there is the issue of sample transportation. And sample transportation will take, for example, how long does your sample take to reach the laboratory, and on what condition is it being transported? Is it transported in a cooler box, for example, or such kind of a thing? Then there is the pre-analytical phase that now when it arrives to the lab, that when we are preparing samples where we look at centrifugation and all the other liquating and pipetting, probably that would be a factor in the pre-analytical phase. So how do we identify and prevent pre-analytical errors? First of all, identify them by the rate of acceptance or rejection criteria. So the higher we reject samples, then we know most of the errors are occurring before the sample reaches the lab. And how do we mitigate this? For example, we would really rely on training and competency assessment on the staff who are doing specimen collection. Then we would provide at least specimen collection manuals or clinician handbooks for that matter. Then probably employ standard operating procedures or work aids or charts where the specimen are being collected. Then we go to the analytical phase. The analytical phase commences when the sample has already been received and registered in the lab, and usually involves equipment, reagents, and human resource. And usually it's towards the competency of the human resource working in that lab. These errors are usually classified into 2 broad terms, either random errors or systematic errors. It has more focus since this is now in full control of the laboratory professionals. And it is important to note that technology has a great impact and has really improved this phase of the testing cycle. And to prevent these errors, usually the laboratory does quality checks, and these are involving IQC, that is daily IQC, internal quality control, and also external quality control, which is also known as proficiency testing. We also do validation and verification of procedures before they are rolled out, and we also do training and competency assessment for the personnel working in the lab. Of course, standard operating procedures is a baseline for all, and sometimes we provide working aids for the same. Errors in the post-analytical phase, it comprises on the steps after the review and the results have been released from the lab. And this covers up to the interpretation of the attending clinician. Critical values or alert values communication falls into this phase, and errors that might occur in this phase usually are the delay in delivery, that is the turnaround times, failure to report or reporting to the wrong healthcare provider, transcription errors, incorrect results, and misinterpretation of the results by the clinician. So how do we identify these? Usually we identify through customer feedbacks and surveys. Also, audit will help us to identify these errors are occurring. And how do we monitor? And also, if we monitor the service delivery charters, we would know if we are achieving what we said we would do, for example, in Kenyatta National Hospital. Then we would also provide tracking tools and checklists that will help us to see whether we are meeting what we said we would do. Communication logs also helps us to do this. To prevent this, basically it's through sensitization and case discussions. So as I had mentioned, this is just a summary in the laboratory testing cycle. And if you can see the error rates, the most errors occur in the pre-analytical phase, and through many studies, it has been estimated that 46 to 68% of the errors occur in the pre-analytical phase. And in the analytical phase, we have about 7 to 13% of errors occurring, and in the post-analytical phase is between 18 and 47%. It is important to acknowledge that within the laboratory where the analytical phase is the lowest errors occur within that phase, but those processes that are outside the laboratory bear the most error rates in the total laboratory testing cycle. Just to summarize is that the highest frequency of errors with high risk for the patients occur in the pre-analytical phase, but the analytical phase is less prone to errors compared with the processes that are performed outside the lab. So just to summarize my talk is we need to understand the laboratory has both ends, either value versus harm of the laboratory testing. We all acknowledge that 40 to 70% of medical decision depend on laboratory testing. But it is important to accept that testing sometimes can cause harm. And in general, according to Epner et al., this could be because an inappropriate test may be ordered, or the appropriate test may not be ordered, or the appropriate test result may not be used properly, or the appropriate test result may be delayed or missed, and finally, the appropriate test result may be wrong or inaccurate. That's all I have. Thank you. Back to you, Mr. Mokaya.
Great, Mr. Kiptim, for that presentation. This is important to know where these errors can occur and the devastating effect. We will move to our third presenter, Dr. Makori Jamen, requisition of blood and blood components. Dr. Makori.
Good evening.
Dr. Makori, are you?
Yes.
Good evening.
Let me share my screen.
Yes.
Just a minute.
Can you attempt a screen share again?
Okay. I hope you can see my screen.
Yes.
Okay. Good evening, everyone. I'm Dr. Makori. I'm a pathologist in the hematology lab and blood transfusion units. I will talk about the requisition of blood and blood products. I will focus more on the blood transfusion unit. Maybe at the end, I will talk about the hematology lab. So to start with, I will talk a bit about the introduction to the blood and blood products available at Kenyatta. Then if you go through a few of the indications for transfusion and required information for requisition, then we look at the way forward. So I believe most of the people on this forum are familiar with the Kenyatta National Hospital, especially the blood transfusion unit, and are aware that these are the products that we find there. We have packed red blood cells, platelets, concentrate, fresh frozen plasma, and cryoprecipitate. We don't dispense whole blood for obvious reason, because we need packed cells and these other components. So there is no need for whole blood, though it still hasn't, there are still indications for whole blood, but there is a better way to go around it for you not to expose patients to the transfusion reactions that are likely to occur when you administer whole blood that contain white blood cells. So starting with packed red blood cells, I would emphasize first on the fact that we only use transfusion for therapeutic use. We know there are so many other uses, even for packed red blood cells, like for athletes. We don't condone that, and we only use for therapeutic reasons. So these packed red blood cells are separated from whole blood post-collection. Then the indication is for replacement when you are dealing with anemia due to various factors, be it hemolysis, be it deficiency in iron or other elements. Blood loss, I'm talking about hemorrhage. When you are dealing with exchange transfusion, the information that will be required on top of what is on the card, we would like to know the exact diagnosis. When most of the people fill up the card, they just write their anemia, but we would like to know more about it because that helps us to project what else will be needed in the short time to come. Let's say if it is a PPH, this is maybe a mother has delivered and she has hemorrhage. If you come to request for blood, tell us there is a PPH. Then we know they want to need only one unit. And on top of that unit, they may need other products because we're likely to enter into a massive transfusion protocol. Then the age and weight. In our hospital, though we are moving from that culture, there has been a habit of writing C for child. We know a child can be 2 months, can be 5 years, can be 13 years, or writing adult. The adults we are still getting, we moved away from the C, but A we are still getting as age, meaning an adult. And it is very wrong because a 14-year-old and a 72-year-old, the requirements are different. And when you are looking at the values given, they may give you a different information. Then the hemoglobin level, the threshold for transfusion for packed red blood cells in Kenyatta is at 6 grams per deciliter. We are talking about NHB below 5.9, but that is in general. We know that in some cases, that has to be higher. Like in neonates, NHB below 7 grams per deciliter, they need to transfuse. We have also other special cases where we need to assess a case by case, but in general, it is below 6 grams per deciliter. Then the information we would like to know is whether you have just given another transfusion. Let's say somebody comes from theater, a patient is bleeding. We need to know when did you last take the other unit. Because if we're giving more units, we understand that we're administering also the anticoagulant and we'll have to neutralize its effect. That is when now we start introducing things like fresh frozen plasma. So please, when you come to BTU and you are asked many questions, be patient. It is for the good of your patient. We all want our patient to come out alive and we are there to support. If you are dealing with an emergency, maybe it is a massive accident and you need uncross-matched blood, there should be a clinician to sign in the PTU. It doesn't make it more dangerous than the cross-matched one because we have a protocol. Within the 15 minutes you have taken blood, you will receive a call either to continue or to come for another cross-matched unit. Because as soon as it is given to you, then cross-matched unit, that same person sits down and starts cross-matching another unit. And even that one to confirm that it is the correct one. Then another product we have is platelet concentrate. These are pooled platelets from different donors. We sometimes also get apheretic platelets that are corrected through apheresis. Unfortunately, our hospital does not have an apheresis machine, which we may acquire soon. But in the meantime, we collect platelets from different donors. Then we create a unit, and that unit is the one we use for transfusion. When you compare with apheresis, where you take a donor knowing the platelet count, if I can remind you, the platelet count for us is 150,000 to 450,000 platelets per microliter of blood. And this is a wide range, but when you need the donor for apheresis, you will take at a minimum of 250. Unfortunately, when you are doing pooled platelets, you could be getting those ones of 150, 170. So the outcome are not likely to be the same. So when we get apheretic platelets, they're usually donated to us from our local institutions that do apheresis. The indications for platelets are mostly thrombocytopenia, but as I said, also as part of the massive transfusion protocol. There are contraindications for platelet transfusion, and this is in case where thrombocytopenia is immune-mediated. Because if the body is destroying its own platelets and you're adding more platelets for different donors, the body will mount more a higher, more stronger immune response, and it will wipe out even the ones that were remaining in the circulation. So there are better drugs to use in that case. And that is why you may be asked if you're coming for platelets, what other tests have been done? Like was a bone marrow done? Has the patient been seen by a hematologist or a pathologist? All those are questions we may have to ask you. The threshold for transfusion is different depending on the situation. For surgeries, for patients going under general anesthesia, it should be above 50. They should have platelet count above 50,000 per microliter. But for those going for central nervous system surgeries or eye surgeries, this goes higher, up to 100. So in that case, even if the patient has a platelet count of 55 and the request for platelets, we are supposed to give them. We won't argue with them because we understand in those areas, there is, by the time the surgeon comes out of the surgical field, they should have achieved hemostasis. So there is no more room to manage the hemostasis. So it is better they enter when they are prepared. And any patient who has platelet count below 20,000, you should be ready that they can have spontaneous bleed. And this includes intracranial bleed that are better prevented than treated. So these are the patients where I should immediately order for platelets. The information that we require, the usual age, gender, weight for the children, diagnosis, the current platelet count, and also, if not fully evaluated, recommended to have a hematologist or a pathologist opinion in that case. The other product that is separated from plasma is fresh frozen plasma. This is separated when blood is still fresh, then it is frozen. This one, we use it a lot and mostly for coagulopathy because it is very rich in coagulation factors, mostly the ones that are vitamin K dependent. If I can remind you, that is factor 2, 7, 9, 10, and also the naturally occurring anticoagulants, protein C and protein S. They are very rich. The FFP has them in plenty. And we also use FFP to reverse the effect of oral anticoagulant. Let's say a patient has been on warfarin and they have been taking the drugs regularly, then they need to go to be taken to theater, or it is a lady, she has forgotten her menses have started. We can use FFP to reverse the effect of the oral anticoagulant. Another area where FFPs can be used is when you are doing plasma exchange in TTP, that is thrombotic thrombocytopenic purpura. You use FFPs. The information required on top of the usual age, weight, diagnosis, coagulation screen, we will require the INR. INR is calculated from the prothrombin time. In this case, whenever the INR is above or equal to 1.5, there is an indication, give FFPs. For cryoprecipitate, this is another component also separated from the plasma. Once the plasma is separated, it is frozen, then allowed to thaw, then the precipitate is collected and frozen again. This precipitate has now more factors than the FFP, and the factor here of interest is the factor 8, factor 12, fibronectin, von Willebrand factor, and fibrinogen. Remember, factor 8 is the one that is deficient in hemophilia A, and that used to be the most common use for cryoprecipitate until we got the commercial coagulation factor 8 that is available in the hospital, the factor 8 and factor 9. Then it is used to control coagulopathies in hemophilia and also as part of the massive transfusion protocol and also plasma exchange. The information required are the same as the ones for fresh frozen plasma, but in this case, we need also to know the values of the activated partial thromboplastin time. This is the APTT, fibrinogen, and the dimers, because we know by the time we are requiring this cryoprecipitate, there is a lot more going on. So you may be asked more information about the patient. What have you been giving? If the patient is bleeding, what is the hemoglobin level? Because we need to go back there. Because by the time we are giving some of these products, it means something is happening. And to counter it, we may need more than the product you are requesting. The common problem we are having is some of our colleagues thinking that you can use this product interchangeably. Somebody comes for platelets, they find that we don't have platelets, they request for FFP. As you have seen, FFP and platelets supply different components. So if you needed to correct thrombocytopenia using platelets, FFP won't help you. It will supply the coagulation factors, but it won't supply you the platelets that you need. So kindly be understanding, and when you are requested to answer some questions from BTU, if you are not aware, refer to the primary clinician who is likely to be in the ward. These are some of the cards. We recognize that the card we are using has very scant information. As you can see, we usually request for the patient's name, the hospital number, age, sex, the ward, the bed, and the reason for transfusion. It is always anemia. It never changes. I haven't seen many people putting something else, maybe leukemia or anemia in such and such condition. It is rare to see that. Then we really don't require that you tell us the patient group, because whenever you request, we always start from the beginning. We start by grouping the patient. This allows us also to confirm our results with the previous one. We do our own ABO and results grouping. Then date of previous transfusion. This is very important, and most of the time it is left blank. It is important because it allows us to know what happened in the last transfusion. Were there any reactions, or how many units did the patient get? We would like to know how long ago. Why are we continuing transfusing? Maybe we are not addressing the cause for that anemia. Then details of any reaction to transfusion. And this is, I would here remind you that we have a form for reporting of transfusion reaction. Our form is detailed. I wish I had a picture of it, but we have it in the blood transfusion unit. The form will guide you as you put everything down, your observation. It will guide you on the samples to take and even how much, the amount you have to take and what you need to carry back to the blood transfusion unit. Then we will take over from there. We may even come to see the patient, but it is very important that we document. Currently, there is documentation online. You can even be anonymous because people fear reporting such transfusion reaction. And I wish to remind you that it is nobody's fault that the patients react to transfusion. But if you keep on reporting to us, it will allow us to see what products patients are reacting to more. Maybe we need to add in an extra step to reduce those reactions. Please kindly be reporting them, even if it is just chills, report them. Maybe the unit has gone on for days. We would like to have that information and document it. Then the degree of urgency, of course, for everybody it is always urgent. Then the number of units required. For children, we prefer that you write in milliliters. So you calculate the total volume, you write for us in milliliters. Then the date required. Of course, these cards come with so many information that are spread all over the card, and it becomes difficult to assess the level of urgency. So when you are dealing with a patient who needs urgently blood like this, I would request you that you inform the staff in BTU. Don't just throw the card and the sample there and go and wait to be called. Please talk to them, tell them, brief them what is happening so that they can give it a priority. We are here to save all our patients. This is one card. This is another card, very, very urgent. But when you look at the information given, this was for exchange transfusion. With very scanty information, but we know where the patient is in the newborn unit. So these are the kind of information we get. As time goes, because we understand this card has been around for many years, we are in the process of adopting a more elaborate card from the Ministry of Health. I can't project it here because the hospital hasn't yet adopted it. But in the process, this other card has more information, it is more detailed, and it will help us to get all the information we require. Talking about information, I would also encourage those who request for hemogram and peripheral blood film, even bone marrow evaluation, to be giving us more information. Sometimes some of the findings you get on the examination may not seem very important to you, but for us, things like presence of lymphadenopathy, hepatosplenomegaly are very important when we need to advise on the next, if you don't get a diagnosis, to advise on the next step in the investigation. Maybe the patient needs a fine needle aspiration from the lymph node. Maybe you need this and that test. That we can recommend when we have all the information. But when you keep on asking you, is there splenomegaly, we are delaying the time for diagnosis and exposing patients to unnecessary treatment that could be avoided. This was my short presentation. I hope I haven't taken much of your time. Thank you.
It was worth it, Dr. Jermaine Makori. Thank you for the presentation. We quickly move to POCT, a presentation by Dr. Kasim Ali, microbiologist, stroke immunologist. Welcome, Dr. Kasim Ali. Hello, Dr. Ali. Dr. Ali? I can't hear him, but he has shared the POCT policy.
Hello, can you hear me?
Yeah, I can hear you now.
Okay.
Go on.
My
please.
slides are moving. Okay, thank you so much.
We cannot, yes.
I will try to rush through my presentation on POCT policy. This is a document that we've been working on at Kenya in Kenyatta National Hospital. This is the outline of my presentation. We'll try and see how we can have this covered in the shortest time possible. Point of care testing refers to the performance of any clinical laboratory test outside the centralized laboratory. So this may be near patient testing, it may be bedside, extra laboratory testing, or even sometimes referred to as doctor's office of clinic testing. This is usually a rapid, timely, and reliable test, and mostly helping you to make quick medical interventions and aiding decision making to optimize treatment. And mostly the role is in reducing delays that may occur in patient care, reducing development of complications and overall hospital stay. Studies have found utility of point of care testing in screening, diagnosis, identification of risk factors, and even in monitoring of patients with non-communicable diseases in low and middle income countries. And it's gained popularity as most of the tests, including studies that have shown that HbA1c and creatinine POCTs were found to be performing comparably well to the analytical laboratory tests on specialized and complex laboratory equipment. Now, the important thing to note is that POCT services were previously accredited separately from laboratory services under ISO 22870-2016 standard, but there was very poor uptake of this standard. Now, in the renewed ISO 15189-2022, the POCT requirements that were in a different standard have now been integrated in the ISO laboratory standard 15189-2022, in the hope that this will improve the uptake of accreditation of POCT services. Remember, laboratories were getting accredited, but POCTs were being left out. This will allow institutions to leverage on existing skills and knowledge in quality management and risk management to increase accreditation scope to include POCT services, as well as guarantee continuous provision of high quality POCT services. Hence, our statement in KNH is that the POCT policy is designed to improve the quality of patient care and reduce the risk of clinical errors while carrying out point of care testing. And KNH is committed to ensuring that this is achieved through proper guidance and oversight from selection, training, quality assurance, monitoring, and evaluation of POCT services. And our overall objectives would be to offer guidance and support to all POCT operators to ensure all patients receive a standard and high quality care, and to ensure that KNH complies with relevant international and national regulatory standards, and to apply principles of quality management and continuous quality improvement for all POCT services. This will require a multidisciplinary committee to offer this guidance and to carry out the governance of the POCT program. And this team will supervise and oversee quality management of POCT services. And the framework generally will look like this, and these are some of the aspects that will be covered moving forward. An important thing is that the POCT program will endeavor to create an updated inventory of all devices and equipment utilized in POCT services in the hospital. You can only imagine the number of POCT equipment in our hospital. They are probably in the hundreds, yeah? This inventory will include the status of the devices, including the quality control performance of those equipment. Then when it comes to procurement, procurement shall be initiated through a formal request by the clinical team. The POCT shall engage the clinical team and the Department of Lab Medicine to establish a justification, a proper justification for the clinical need and develop appropriate specifications. And together with the supply chain management, the POCT committee will oversee procurement of appropriate devices. And considerations during this process shall include assessment of the benefits to the patient, the benefits of our alternative tests that are available in the clinical laboratories, and the evidence base of the performance of such POCT tests, ICT and end-to-end connectivities, as well as cost effectiveness. Then once procured, it's going to be a requirement that all POCTs shall have standard operating procedures wherever they may be stationed or operated from. These equipments shall be required to have calibration and test method verification, internal quality assurance, internal quality control processes being performed for each POCT device. You can imagine that our hundreds of POCT devices in the hospital are not having internal quality control, done or performed. So we cannot really rely on the results that are being generated from this equipment. Enrollment into external quality assurance is going to be a requirement, and reporting of these results and even seeking connectivity, so that then we make sure that there's evidence that results were run on an equipment, the results were documented. If there were critical results, that these critical results were communicated appropriately and in a timely manner. All vendors shall be required to have a service level agreement to ensure that appropriate maintenance of the POCT devices is maintained. And the institutional framework shall include the administration of these POCT services, the structure and organizational chart, a POCT committee that shall be in place, and the roles and responsibilities of each. So this is a framework of the organizational chart, basically a representative from all the healthcare units that we have: physician representative, critical care, nursing, biomed, pharmacy, quality department, ICT, lab medicine. And hopefully all these under the leadership of the POCT committee chair and with the help of the POCT coordinator shall manage this program. Importantly is the role of the POCT coordinator. The POCT coordinator shall be a qualified and experienced lab technologist who shall serve as a contact person for all aspects of POCT programs. And the roles shall include participating in developing specifications for POCT, participating in technical evaluation in the procurement of devices and equipments, monitoring and evaluating and reporting on the performance of the POCT devices, including status of training and competency of the people who are running or operating this equipment, the IQC, internal quality control, and the external quality assurance performance, and the equipment status and the clinical errors and so on that are coming out of these equipment, developing SOPs and working aids for the POCT programs, and preparing and coordinating and reviewing the user training and competency assessment plans. As well, it's expected that this is going to be the person who will be doing daily work of implementing some of the quality programs, including assisting the users on matters to do with quality, including IQC, lot-to-lot verification, TMVs, participation in EQA. He's going to be the custodian, he or she will be the custodian of all inventory and records, and will develop annual projection for POCT devices, and will be the advisory to user departments whenever they need any sort of help. Another important person is the unit manager. Every unit that is carrying out POCT, it's expected that the unit manager shall have some roles in this program. And this will include supervision of all personnel and POCT services that are being provided within that unit, ensuring that POCT services are carried out only by personnel with the appropriate qualification and training. The time of asking anyone who's in the ward, whether they are a student or not, to go and run a test just because it's available within the ward, without making sure that that person is competent enough to run that test, are gone. And that's the requirement is that the unit managers shall ensure that only trained and qualified personnel are operating this equipment, and ensuring that they carry out training and competency assessment and maintain up-to-date qualification and competency assessment records, as well as maintaining testing, IQC and EQA and maintenance records for the equipment, and ensuring that all critical results are performed. Thereafter, the policy will provide a detailed M&D framework that may require quarterly or annual reports of POCT coordinator or the committee to the management. We'll see some of the KPIs that may be recommended, and importantly, maintaining some of the records that previously have been missing. When you go to the areas where these POCT equipments are being operated, there's literally zero record in regards to the equipment or when they were procured and their status in terms of maintenance and in terms of performance. Some of the KPIs will include pre-examination errors or instrument alerts that will have to be recorded, analytical errors that have been reported, critical result communications, and IQC performance and EQA performance, and if there are new POCT services that are being introduced. So I want to stop there due to time, and we'll take any questions if and when they come up. Thank you.
Thank you, Dr. Ali. Our next presenter is Dr. Ndiangui, who will give us a sensitization on Clinician handbook. Your time is very much limited, Dr. Ndiangui. We hope you will be able to bring out the salient points in your presentation. Welcome, Dr. Ndiangui.
Thanks. I think I may just take 5 minutes. I think it's just for just a minute. Sorry, I think I've shared the wrong one. Good evening once more. Yes, I'll try to move as fast as possible because it's just a highlight, nothing much, and so that we have time for reaction to all the presentations. So, laboratory clinician handbook. So, as a department, we are required to communicate relevant information to our clients. That's both the patients and clinicians, as to inform them of our services. This is part of the requirement of the ISO standard 15189/2022 that we inform or we have interaction with our clients, inform them of our services. And this is just a bridge of the relevant information. So in this clinician handbook, we should have at least the location of the laboratory, where are they, to explain, the operating hours and contact information, the procedures for the requesting and the collection of samples, the scope of laboratory activities and time expected of available results, that's the TAT, the availability of advisory services, where can you, number you could call to seek more or information regarding the test or the services. Then you also, information of the requirement of patient consent. The fact you are taking a patient sample, you are performing a procedure, you require consent, sometimes oral, sometimes written, but that procedure has to be indicated in the manual. Then other issues that should be communicated is factors known to significantly impact the performance of the examination and the interpretation of the results. Then the laboratory complaint process. How do you interact with the client in terms of if there's a complaint or a feedback, then it should be communicated clearly how it should be handled. Also, specimen rejection criteria. You explain to the patient or to their client what is the criteria that we are rejecting the specimen. If, for example, you are collecting blood, then you have what is the procedure? The vacuitainers, including colors and the additives that may be having. Which order do you draw blood if you have several tests that are being done and requiring several sample containers? You are doing hemogram, you are doing UREs, which require different additives or no additives to the vacuitainer. Then also you need to communicate about different values, but all this is in terms of, it is in a more abridged version. And all this information given usually is in detailed, mainly in the what we call the specimen collection manual, which is also available in the lab as a standard operating procedure. So this all information is available in that procedure, in that manual. So just to mention our laboratory sections, which are in, they are mainly microbiology, renal, the HLA lab, immunology, critical care unit. We have the blood transfusion, parasitology, KPCC and COC, and comprehensive care center. You have hematology and as well as histopathology section. These are the laboratory sections that we have in the hospital, which are contained in the manual. So by standard, all this clinician manual should be reviewed regularly, and the currently in use was effective date was in March 2023, and the other one it's due for review this year or at the end of this year. So I just want to show, I don't know whether the link will open, but so we have this is the clinician handbook which we have is currently in circulation, and you have it's in soft copy, and also there's a hard copies which are in the sections, and I think was circulated in the ward. So they contain all that information, some of which, as I said, are also contained in our laboratory manuals. And because of time, and I don't think I need to go details to it, but the purpose of this presentation was to make our clients and our patients aware of it and its use in the hospital. So back to you, Mr. Mukaya.
Thank you, Dr. Ndiangui. We now move to the plenary, where we'll take any concern, any comments or clarification that participants may want to get from the panelists. If there is anybody who want to get some clarification or a comment on what has been presented, you are welcome.
Maybe I could take a question from the chat box that has been raised by one of the participants. It says, good afternoon, kindly expound on the self-request aspects. Is it legal or acceptable, especially to the patients who visit standalone laboratories or undergo routine monitoring tests? I think this arises from the issue of request form that I presented, was the first presentation. And I don't think there's a yes or no answer, but maybe what I omitted in my presentation is that there is room for, as first talked about the request form, there is room for oral request in circumstances in a hospital setup, but that should be also be recorded and followed up with a request form being written. So a clinician may ask a request for a sample, to maybe a sample which was collected earlier may request more further whether the menu can be expanded, but that should be recorded. In a standalone laboratory where a patient walks in and wants to be done a request, I believe if the patient is requesting, then it should be accompanied by whoever is being requested, then requested to do should put down that information in a request form and before the test is being performed. Of course, I'm alive to the fact that there is a lot of POCT testing, which probably even doesn't need a request form for that matter. But yes, I don't have a clear answer whether yes or no is it legal or not. But I hope I have been able to at least to attempt to answer that question.
Okay, I've seen in the chat box somebody asking whether we can circulate the document. The document is already in circulation. We distributed to the clinical areas last year, and the section in charges have access to it, and if anybody is wishing to have a reference to it, it can be provided. Any other concern? I've seen in the chat there is a question by Duncan Odera. Mr. Peter, kindly clarify if histopathology and cytopathology are one subspecialty or different subspecialties.
Maybe I could take it, Mr. Mukaya.
Yeah. Yes, Dr. Ndiangui.
Yes, that's a good question. Whether histopathology and cytopathology are different subspecialties. Basically, we have in what we call surgical pathology. Surgical pathology covers cytology, histology, and sometimes also forensic medicine. That is area to do with postmortems. Yes, cytopathology is under surgical pathology, and you can consider as a subspecialty because you can do cytopathology alone as a degree or at that level. So cytopathology is part of surgical pathology which follows under which encompasses histopathology, cytopathology, and also forensic medicine, forensic pathology.
Okay. There is a general concern from the chat that the slides be shared. We will liaise with the research who are in charge of this program and see whether they can share the slides to those who are requesting. I've seen quite a number. Any other concern? I'm going through the chat and the requests are just the same.
Okaya, there's a query I think maybe targeting the POCT presentation. It says, what if there are no facilities for that separation, temperature control, centrifuge? So, I mean, essentially POCTs are designed to bypass some of these preparations, yeah, that may include, separation of the samples, yeah. Most of them are designed to use whole blood, yeah, in a way in which it can be used or even use, without the need for separation, without the need to process or analyze plasma or serum. So there may be no requirement in most cases. So again, but this is part of what, before you procure, you'll be going through. If there's going to be such a requirement, then the question will be, why then provide such a service? So it's important that that is captured during procurement. Thank you.
Okay. There is a comment there. The clinical information of the patient has always been a problem. Most of the doctors do not indicate that aspect, which is very key when analyzing. What can we do? I think this has been the gist of this presentation, and I hope that has been handled because this was a presentation meant to correct that anomaly. There is another one. What is the criteria in deciding who takes which sample? I have noted some samples being collected by lab personnel, some by nurses, then others by medical officers. Sometimes there is confusion on who should pick the sample. Sample collection is only undertaken by a clinical personnel. So all the people you mentioned here are actually authorized to collect specimen with the proper coordination. Maybe when you talk about confusion, that amounts to just coordination. But I don't see any harm for a medical officer, a clinical officer, or a nursing officer collecting a specimen, because even nursing officers have done phlebotomy services and they're competent in that area. They're asking where they can download the clinician handbook. This seems to be, this can only be, I think, operational within Kenyatta National Hospital, unless otherwise guided. Any query, any concern other than the chat box information we are giving? There is one who is saying, good afternoon. Is it possible to have the notes on blood products and errors in the laboratory for reference? As I've said, this one can be handled by the research team that is handling this platform. Because the presentation which has been given on blood products can be converted and given, shared out with permission from research. Thank you. On CPD, unfortunately, this platform has not been accredited to offer a CPD for laboratory technologies, but we are still working in conjunction with KMLTTB to onboard this platform to be offering CPDs. So that is a matter we will take up and continue handling with the KMLTTB. Due to time constraints, because we were supposed to be over by now, unless somebody has a burning issue, I would wish to hand over to research and programs. And before I do that, I would just end by saying our desire as a department is to provide effective and efficient services to our customers. This can be achieved through constant interaction that emphasizes on honest and prompt feedback. And I believe today's presentations and interactions have worked toward that end. Thank you. And research and programs, are we there? Are you there? Okay, we have come to the end of this program. Thank you for finding time to be with us. Thank you and God bless all of us. Asante.
Thank you. Nice evening.
Okay.